All right, so chapter 39 talks about plant responses. And again, this is a little bit about some of the stuff that we talked about in the last chapter, chapter 38, dealing with short and long day plants. And basically, what it's talking about is the reproduction in flowering plants is influenced by both the biotic and abiotic environment. So, if we look here at the responses to stimuli, of course, we know that plants are sensitive to a wide range of stimuli, and it's what's going to elicit some sort of a response. And what they're going to use is a signal transduction pathway. Excuse me, and we've talked a little bit about these signal transduction pathways, and what they do is there's some sort of an activator on the exterior that's going to be received by the plant, the cell, whatever really type of organism it is. The same general kind of mechanism is involved, and these proteins are going to respond to a certain kind of stimulus, and that's going to result in a series of changes, a cascade of changes that's going to lead to some sort of a cellular response. So we have these second messengers that we've talked about, and these are small chemicals that are produced on the inside of the cell, and what they're going to do is they're going to amplify the signal. So for instance, like a small amount of hormone will yield a large response in the organism. And one signal, for instance, one signal molecule can give rise to hundreds of specific types of enzymes that's going to lead to a rapid amplification of the signal, and again a big cell response. So if we're looking here, for instance, with light, the light is going to activate a second messenger, and you're going to get some protein kinases that are going to be activated, and then you're going to have a response that's going to be generated from this, and you're going to get more of these protein kinases activated, and these things of course are going to lead to changes in the nucleus of the cell. And for instance, with the case of plants, if we want to talk about, like, if you've ever left a potato in the cabinet for a long period of time and it gets these long kind of white growths that are off of it, those are the shoots. They're essentially searching for light, but as soon as you give it a little bit of light, those things start to green up. And if you think about it, it doesn't make any sense to make any chloroplasts or chlorophyll or waste all of the energy making those types of things if no light is available. So all of the structures really will be there, and then as soon as you give the plant some stimulus in terms of light, the nucleus is going to start being stimulated to express the genes that make chloroplasts, chlorophyll, those types of things. And that process is called greening or de-etiolation. That's the process that we're talking about. And then the plant's going to start to grow and mature. So one of the responses here, the signal transduction, again, of course, is going to lead to a variety of changes within the cell, and that's what's going to lead to the changes that you see in the plant. And two of the main mechanisms that these responses occur is you're going to stimulate either the transcription of the messenger RNA, which of course is going to give rise to proteins, or you're going to activate some existing enzyme molecules, or perhaps both. So when you're stimulating the transcription of the messenger RNA, this is called transcriptional regulation. In other words, you're regulating the amount of transcription that's occurring. And you've got these transcription factors that are going to bind directly to the DNA molecule, and this is what's going to control the transcription of specific genes. And then if you're activating certain enzyme molecules that are already there, this is a way that you can get hundreds of second messenger molecules leading to the activation of hundreds or thousands of enzymes. So you can see, if we look right up here, we've got the activation kind of stimulus right there. That's going to lead to the activation of a protein that's embedded in the membrane, and then that is going to give rise, this particular instance, it's going to give rise to a bunch of these cyclic AMP molecules. Each cyclic AMP molecule is going to stimulate, in this case, this example here, you've got a protein kinase A, so you can see each one of these is going to give rise to more pKa and more and more and so forth, and then each of these is going to give rise to lots and lots of protein kinases, which is going to give rise to more. And again, you can see all along the way that this small signal right here is amplifying itself all along the way of the internal portion of the cell, which is going to lead to a large response in either the cell or the organism, or both. Okay, and again, this is just kind of looking at back here what we were just talking about. This is the way this kind of thing is playing out in the example that we looked at earlier. Okay, so now just a couple of the things. It'll serve a little bit as a review from the last thing that we went over in terms of light causing the change in the phytochrome. This phytochrome is going to lead to an increase in this, cyclic AMP or cyclic GMP in this case, some calcium molecules getting in and activating all of these protein kinases. This is the sort of thing that we were talking about earlier. And then more specifically, this is the way in which it occurs. Like looking at this, you can see up here, again, here is the phytochrome that we were just talking about. And looking in this example, here's the phytochrome again, And what's happening is we have light that's interacting with it like we're seeing here. And this light is giving all of these responses over here that we're talking about. Okay, so that's how the change in phytochrome is really leading to these changes that we've been discussing. Okay. So these protein kinases, these things become activated by a series of molecules, cyclic GMP, calcium, and what they're doing is they're phosphorylating and activating other types of enzymes. And these enzymes can then be used to either stimulate and kickstart a process, or they can shut it down. And just depending on how they're being used, what type of proteins you're talking about, but that's how they're essentially being regulated. And then what happens is once you're affecting the transcription of these genes, the enzymes can then, of course, make proteins to elicit the response that we're after. Okay, and the way in which phytochrome is actually occurring, you have a mechanism that's involved in what you're getting is the new development of either a new flower or a new growth or something like that is going to have the activation of a series of reactors that are involved in either turning the genes on, turning them off, getting the plant to flower, or for instance not flower, whatever the case may be. And we've talked about light. This is being very important in the growth and development of plants. You get photomorphogenesis. This is where the light is going to have an effect on the plant's morphology. In other words, whether it's green, whether it's going to grow leaves, whether it's going to grow a shoot, whatever the case may be. And the plant's ability to detect this light, the length of the day, the length of, more accurately, the length of the night, is how the plant is going to measure the passage of the seasons and know when to go dormant and of course when to grow and flower and those sorts of things. Okay, so photoperiodism, this is something we talked about in an earlier lecture, and this is the really plant's way that it's going to have a change based on the length of the day or the night, and it's actually the length of the dark period. So again, now this is just going to kind of get into some reviews. The three general varieties of the plants, we talked about long and short day plants, which are actually long night and short night types of plants, and then there's just day neutral. They're not responsive to any of the photoperiod. And again, short day plants, this is what we said were the long night types of plants, and these are going to often flower late in the summer, fall, or winter, as this light period is going to be shorter than 14 hours. So now, in other words, we're starting to lengthen this nighttime period. These long day plants now, they're more accurately referred to as short night plants, and these are going to flower when the light period is longer than 14 hours, or you're starting to get a shorter dark period like we've seen. Okay, and then day neutral, these are things that are unaffected by the photoperiod. All right, so as we said, some of these experiments began in the 1940s with the USDA, and what they were looking at is the length of the days and the nights and how these things were kind of affecting the way in which the flowering was occurring. And what they found was they could break up the photoperiods with light and affect break up the dark periods, I should say, with a pulse of light and affect the plant. But if they were to break up the light period with a brief period of darkness, they didn't find anything. Okay, and we talked about the long day and the short night, or the short day and the long night, and how these things are essentially working opposite of one another. And what they found, of course, was that the red light, again, was the most effective at disrupting this photoperiod. And the phytochrome, this was the pigment that was involved in measuring that photoperiod. And when they were looking at this, they looked at seed germination and flowering. And some of the things, again, that we talked about here were when you subject these plants to a specific color of light, either red or far red, this was what was going to affect the germination as well as the flowering, as we'll also see. And we looked at this picture here, and we were just basically talking about how the red light is the activator, the far red light is the inactivator, and what matters is the last color that the flower receives. In other words, if the last color is the red kind that's going to turn it on, you're going to get germination. And if the last color is the far red and it's going to turn it off, you're not going to get any germination. So those are some things that we covered. And then we went through here how this phytochrome is going to oscillate back and forth between the two forms, which are either turning the processes on or turning them off. And the question that we answered was how the plants did this in nature, and we looked at keeping them in the dark and how they're synthesizing the PR, which is going to be able to absorb the red light and kind of turn this on. this PFR gets formed and then very slowly is converted back to the PR, and that's the adequate amounts of the PFR what was going to trigger the germination and the flowering. Okay, and these two colors of light, of course, we said affect one another by flipping these things on and off. And again, if you look over here, here's our short day or our long night plant. This is not a long night, so we don't get flowering. This is a long night, so we do get flowering. Okay, and then over here, this is a long night, but we break it up into two short nights, and then this here is going to mimic that there, and we're not going to get any flowering. Okay, and then the opposite is true over here. This is the long day, or rather the short night. Here's a short night. Okay, we get some flowering. Here, this is a long night, of course, no flowering. And then we've got a long night, but we break it up. So it's basically like two short nights. So this looks very similar to that. And we get the flowering here. Okay, and when all this picture is over here, all this is showing you is the two kind of overlapping one another. Okay, so there's a lot of other stimuli other than light that are going to affect the plant growth. We talked about some of the things in an earlier lecture with gravity, certain mechanical stimuli, environmental stresses, things like that. So there's a wide variety of things, but this general kind of example kind of explains how these processes occur. So if you got any questions, write them down and we'll talk to you soon.